Okay, so in today's class, we are going to discuss about the uh, prophase one of meiosis. So the prophase one is typically longer and it is more complex stage than other cell division. So the prophase one is divided into various stages based on the chromosomal behavior. So it has five phases, that is leptotin, zygotin, pachytin, diplotin, and diakinesis. So you can remember it by the mnemonics LZP D square. So the various stages are leptotin, zygotin, pachytin, diplotin, and diakinesis. In this slide, you can see a brief introduction or summary of what exactly takes place in each stages is given. In the first stage, what you notice is in the leptotin, so you notice that there is going to be the duplication of the duplicated chromosomes, they start to condense further. Okay, and in the zygote stages, the synapses begin, that is pairing up of homologous chromosomes takes place, and some of these are held together by the synaptonomal complex is also formed, and these homologous chromosomes are held together. Okay, that is one aspect that you should also remember about. Then the next stage, that is in the prophase one pachytin stage, you notice that synapsis is completed and crossing over commences. That is non-sister chromatids of homologous chromosomes, they are going to cross over. So that is taking place in the pachytin. The synaptonomal complex is completed and the uh, crossing over is taking place. So in the diplotin stage, what you notice is you are going to see that the synaptonomal complex is going to uh, disappear and chiasma is visible. In the diplotin stage, the chiasma, the synaptonomal complex, they are going to disappear and there is going to be the crossing over of non-sister chromatids, which is visible as a X-shaped structure. The point at which they have crossed over is a chiasma. So the chiasma is visible at this particular diplotin stage. Diakinesis is terminalization of chiasmata, and a fragment of maternal gets attached to paternal and a fragment of paternal chromosome gets attached to maternal chromosome. So there is the terminalization of chiasmata takes place and a bivalent body is prepared for the metaphase stage. Okay, so these are the synopsis of the entire prophase one. So we'll discuss one by one each and every stages of it in detail. So leptotin, it is also called as leptonema. The stage is also called as leptonema. The chromatin fibers become long and slender. So you can notice that the chromatin fibers are becoming long and slender chromosomes. The nucleus enlarges. So that is what is going to take place in the key changes in leptotin or leptonema is, so the chromatin fibers become long, slender chromosomes and nucleus enlarges. The nucleus is enlarging. In zygotin or zygonema, so chromosomes become more condensed. Okay, the coiling up of chromatin material takes place. The chromosomes becomes more condensed and similar chromosomes start pairing together. That is chromosome 1, maternal and paternal, they come together. Chromosome 2, maternal and paternal, which are similar chromosomes, they start pairing together. Jodi agate alli. So you can notice here the blue colored and red colored one. Okay, so they are going to, similar chromosomes are going to start pairing together, and that pairing together we call it as synapses, and they are going to start pairing together with the help of a complex structure which we call it as synaptonomal complex. So they are going to start pairing together similar chromosomes with the help of a complex structure called as synaptonemal complex. So the paired chromosomes or homologous chromosomes, each pair of homologous chromosome, we call it as bivalent. So each pair of homologous chromosome, like chromosome 1 pair, that is maternal and uh, maternal together, we call it as a bivalent. Each pair of homologous chromosome is called as bivalent. So the paired chromosomes, homologous chromosomes, which have come together and they have paired, we call it as homologous chromosomes. Each pair of homologous chromosome, this pair, complete pair, is called as bivalent. Okay, so the pairing up of chromosomes takes place, the synapses with the help of a complex structure called as synaptonemal complex. So there is a formation of synaptonemal complex which holds together this pair of similar chromosomes, which we call it as homologous chromosomes. Each pair, this two of them together, each pair we call it as bivalent. Each pair of homologous chromosome is called as bivalent. So in pachytene or pachynemia, so in zygotine or zygonema, we found the formation of synaptonemal complex and pairing up of the homologous chromosomes takes place. In pachytene, it is a comparatively longer phase. So the bivalent chromosomes split into similar chromatids. So the bivalent chromosomes split into similar chromatids. This stage is called as tetrad. So the bivalent chromosomes, which are held together, so they split into chromatids, and this stage is called as tetrad. So the appearance of recombination nodules, they are going to appear. Can you notice here the recombination nodules? So the recombination nodule occurs, it leads to recombination of genetic material. So the recombination nodule appears, which leads to the recombination of genetic material on the homologous chromosomes. See the same chromosome having this chromatids, we call it a sister chromatid. So one of this blue colored chromatid and one of this red colored chromatid of the homologous pair, we call it as non-sister chromatids. So this crossing over occurs always between the non-sister chromatids. So, and this crossing over leads to genetic recombination of genes from paternal to maternal chromosomes, and maternal to paternal it takes place. So recombination, genetic recombination or the recombination is completed by the end of pachytene stage. So pachytene stage is the third phase. So it is a comparatively longer phase. Pachytene stage is also called as pachynemia. So the bivalent chromosomes, they split into similar chromatids. So the bivalent chromosomes, they are going to split into similar chromatids, which we call it as sister chromatids. This stage is called as tetrad. So the appearance of recombinant nodules at which crossing over occurs between two non-sister chromatids of homologous chromosome, there is a crossing over occurring. So this leads to recombination of genetic material on the homologous chromosome. That is a fragment of paternal chromosome gets attached to maternal, maternal to paternal. So this we call it as the recombination of genetic material is taking place on the homologous chromosomes because of the crossing over. So the recombination is completed by the end of pachytene stage. That is the third phase. In the diplotin, so the key features that you notice in diplotin is the synaptonemal complex, which was holding the chromosomal homologous chromosomes. They are going to dissolute. Dissolution of the synaptonemal complex occurs. That is, they were held together homologous chromosome by the synaptonemal complex. That synaptonemal concept, it dissolves, and now the homologous chromosomes are held only at the chiasmata structure, and these chiasmata are formed by this X-shaped structure of non-sister chromatids, which we call it as chiasmata. So the recombined homologous chromosomes of the bivalents, they separate from each other, that is the synaptonemal complex, except at the site of crossing over. See, everywhere they are got separated except at the site of the crossing over. So this X-shaped structure, we call it as chiasmata. In oocytes of some vertebrates, the diplotin lasts for many months or many years. So in oocytes of some of the vertebrates, the diplotin stage of prophase 1, it lasts for many months or years together. Okay, so remember about the diplotin stage where there is dissolution of synaptonemal complex, formation of chiasmata, wherein you notice the X-shaped structures are called chiasmata. In prophase one, what else you notice is the fourth stage diakinesis. So diakinesis, last stage, that is the fifth stage, that is diakinesis. During this diakinesis, there is the terminalization of chiasmata, the X-shaped chiasmata which was formed. Now they are going to terminalize, they are going to get out from this chiasmata, and you find that a fragment of paternal will be attached to maternal, maternal will be attached to paternal. So the terminalization of chiasmata takes place. The chromosomes are fully condensed. Okay, the meiotic spindle fibers originate from the poles and to prepare the homologous chromosome for separation, and nucleolus and nuclear envelope, they disappear. So the nucleolus and the nuclear envelope, they disappear as is seen in all the prophase stage, but this is noticed in diakinesis. Excuse me. So now what you notice in diakinesis is terminalization of chiasmata. So that terminalization of chiasmata takes place. The chromosomes are fully condensed. The meiotic spindle fibers originate from the poles to prepare the homologous chromosomes for separation. So the homologous chromosome, chromosome 1, maternal and paternal will be separated and they move towards the opposite poles, which we notice in the anaphase stage. So they are preparing the spindle fibers originate from the poles to prepare the homologous chromosome for separation. Simultaneously, there is disintegration of nuclear envelope and nucleolus. So both of them are going to disintegrate. Okay. So this is about the prophase phases. So important stages that are taking place in the entire prophase. You can summarize it. So there was the LZPD square. Zygote is where there is a pairing up of homologous or similar chromosomes, and we call this as synapses. And these synapses, homologous chromosomes, they are held together by synaptonemal complex. Okay, that is what you noticed in the zygote. In packetine stage, there was the crossing over of non-sister chromatids. So there is you can notice that uh, there is an egg-shaped formation of the chiasmata. Okay, synaptonemal complex, and they form the bivalent of the homologous chromosome. Then it is longer phase comparatively. So the bivalent chromosome split into similar chromatid. So this stage is called as tetrad. The appearance of recombinant nodules at which crossing over occurs. Okay, the number of recombination nodules depends upon the length of the chromosome. So it leads to recombination of genetic material on the homologous chromosome. Recombination is completed by the end of packaging, that is genetic recombination. So diplotin, there is a dissolution of the synaptonemal complex. The recombined homologous chromosomes of the bivalents, they separate from each other except at the site of this crossover. This X-shaped structures, crossovers, which is X-shaped structure, we call it as chiasmata. So in oocytes of some vertebrates, the diplotin stage lasts for many months or many years. So diakinesis is terminalization of chiasmata. We call it as the important stage in diakinesis. The chromosomes are fully condensed. Okay. The meiotic spindle fibers originate from the poles to prepare the homologous over chromosomes for separation. The nucleolus and nuclear envelope, they disappear, as is the case of all the prophase stages. Now, metaphase one, you know that in metaphase one, the chromosomes are arranged on the equatorial plate. So the spindle formation is noticed. Okay, so the microtubules from the spindle attached to the pair of homologous chromosome. So here you can notice in the metaphase one, spindle formation is complete. So the asters move towards the opposite poles and spindle formation is complete. And you can notice that the spindle fibers are attached to the chromosome at only one end of the centromere. Uh, okay, so from both the poles, the spindle fibers are attached to the one of the chromosomes. Okay, it is not the opposite poles of it. And even the arrangement of chromosome, if you look into it, the alignment is different from that of metaphase. So the homologous chromosomes are separated. So the microtubules forms the spindles which attach to the pair of homologous chromosomes. So out of four chromosomes, two moves towards one pole, the other two moves towards the opposite pole. So there is no longitudinal division of centromere and there is no separation of sister chromatids. These are the important points that you should notice in metaphase one, which is completely different from that of mitosis metaphase. Okay, so there is no longitudinal division of centromere and there is so there is separation of chromosomes, homologous chromosomes takes place. So and you can already notice the haploidy is taking place. Okay, so these are things that you have to remember in metaphase one. So the microtubules from the spindle attached to the pair of homologous chromosomes. So the homologous chromosomes separate while sister chromatids remain associated at their centromere. So there is no splitting up of centromere, neither there is separation of sister chromatids. Okay, that is what they are talking about. So in telophase 1, the nuclear membrane and the nucleolus reappear and the two haploid daughter nuclei are formed. So this is called as dyad. After this, cytokinesis may or may not occur. See, the nuclear membrane and nucleolus reappear and the two haploid nuclei are formed. This is called dyad. So where there might be a cytokinesis or there might not be a cytokinesis. So after a short interphase, it is followed by meiosis 2. The short stage between two meiotic division, we call it as interkinesis. So there's a short interphase which is followed by meiosis 2. This short stage between meiosis 1 and meiosis 2, that short phase between two meiotic divisions, we call it as interkinesis. So DNA does not replicate under in the interkinesis stage. So there is no replication of DNA in the interkinesis phase. So the telophase one is completed. So the nucleus is formed. They might get separated as two daughter cells, or the after the formation of nucleus, they might be without cross-wall formation. So that is what you have to remember. Okay. Now we'll go for the meiosis 2. So meiosis 2 is also called as haploid mitosis. So it is similar to mitotic cell division. So now already in the dyad, haploid set of chromosomes are formed. Now the meiosis 2, it resembles the mitosis. That's the reason meiosis 2 is also called as haploid mitosis. Okay, it has four phases, meiosis 2. Already you know that they have two cells or they might be having two nucleus if there is no cross-wall formed. So the prophase 2, metaphase 2, anaphase 2, and telophase 2. These are the stages. In this slide, it is a very much what you call it as a summary of this entire process. In the prophase 2, there is disappearance of nuclear membrane, there is disappearance of nucleolus, Golgi complex, and endoplasmic reticulum. Also, the asters, the centrioles are going to move towards the opposite poles and they form asters with microtubules to which the spindle fibers are attached. So these are the stages that are taking place in the prophase 2. In the metaphase 2, the chromosomes are arranged on the equator. So we call it as metaphasic plate and the spindle fibers are attached to both from the opposite poles, the spindle fibers are attached to both the centromere of the chromosomes. They are attached to both the sides of the centromere of the chromosome. It is similar to the tug of war. So the two sister chromatids, they split at the centromere longitudinally, and the chromosomes, single sister chromatid, they move towards the opposite poles. So that is happening in the anaphase stage. So the splitting up of the centromere longitudinally and the separation of the sister chromatids is taking place in the metaphase 2. So these metaphase 2, it leads to anaphase where the chromosomes with single chromatids, they move towards the opposite pole. And in the telophase 2, there is a reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies. So each cell divides into two daughter cells which are haploid. So the second cell also divides into two daughter cells. So totally from meiosis, so four haploid daughter cells are formed at the telophase stage. So in the meiosis two, if we understand the prophase, it is initiated immediately after the cytokinesis. The chromosomes again becomes very compact. The nucleolus and nuclear membrane disappear in both the nuclei. So there are two daughter cells, dyads. So the chromosomes align at the equator, and the microtubules from opposite poles of the spindle, they get attached to the kinetochore of the sister chromatids. So in the anaphase 2, the simultaneous splitting of centromere of each chromosome, so which was holding sister chromatids together. So thus they move towards the opposite poles of the cell as single chromatids. The two groups of chromosome once again get enveloped by the nuclear envelope, nucleolus. Cytokinesis follows, resulting in the formation of tetrad of cells, that is four haploid daughter cells. So this is the overall step. So prophase 1, so then there is crossing over, genetic recombination would have taken place. So the in the metaphase 1, the chromosomes, homologous chromosomes are separated and they move towards the opposite poles. In anaphase 1, the uh, chromosomes have become haploid, you can notice that, and they move towards the opposite poles. Telophase 1, there is a reappearance of nuclear membrane and nucleolus, and now this dyads, what we call it as, or the two daughter cells, they undergo meiosis 2. Meiosis 2 is also called as haploid mitosis because the division is similar to mitotic cell division. So now they enter into, both the daughter cells enter into prophase 2. During this prophase 2, what is happening is, so there is the chromosomes, they are going to start to pair up. So the synapses, the chromosomes are going to be arranged much more compactly arranged, and you can look the structure called as chromosomes. The next step in metaphase, the chromosomes are arranged on the equatorial plate or metaphase plate. So now the asters with the spindle fibers, they are attached to both the sides of the centromere. It is like a tug of war, and the centromere splits longitudinally, separating the sister chromatids, and these sister chromatids moves towards the opposite pole. Single sister chromatid with centromere moves towards the opposite pole. And now this karyokinesis is followed by cytokinesis. There is a reappearance of nuclear membrane and nucleolus. Then there is also reappearance of the Golgi complex and endoplasmic reticulum. So in telophase 2, you totally find four haploid daughter cells, and they are genetically recombined also. Are you able to hear me? Or for some of you, if you are having audio break, um, so you should, yeah. See, one is internet connection. You have to check it with, if there is a audio break, there is internet connection would be there. So data might have got exhausted. That is also one of the reasons for it. So what you can do is I'm already recording it and uploading it also. You can again listen to it. Okay. So anyway, remember about this complete synopsis of meiosis. So how there is finally the formation of four haploid daughter cells at the end of two meiotic cell division, meiosis one and meiosis two. Okay. So the overall step of meiosis one. So the parent cell undergoes interphase. There is a formation of duplication of the DNA takes place. So in the prophase one, which is an elongated phase, so the pairing up of homologous chromosomes, then there is crossing over, chiasmata formation, then splitting up of this chiasmata and genetic recombination would have taken place in the prophase one. So now the chromosomes are genetically recombined. This prophase one enters into metaphase where the chromosomes are arranged at the equatorial plate. So the homologous chromosomes are separated and they move towards the opposite poles. So there is no splitting up of centromere longitudinally and there is no separation of sister chromatids. Rather, the homologous chromosomes they move towards the opposite poles in the metaphase one. Now, the chromosomes are arranged at the opposite poles because of the condensation of the spindle fibers, and then it enters into telophase, where there is a reappearance of nuclear membrane and nucleolus along with endoplasmic reticulum and Golgi complex. And you can notice that two daughter cells are formed, and each one of them is haploid. So they might form two daughter cells. So the karyokinesis might be followed by cytokinesis, or the nucleus again undergoes meiosis 2. Okay, in the meiosis 2, it is also meiosis 2 is also called as haploid mitosis. So what is exactly going to happen in meiosis 2 is they enter into prophase 2. Two cells will be there, two daughter cells. So they undergo metaphase 2, wherein the chromosomes are arranged on the equatorial plate, metaphase 2, and the spindle fibers are attached to both the ends of centromere, and there is a longitudinal splitting up of the centromere and the separation of sister chromatids, which moves towards the opposite poles during anaphase. In the telophase, there is reappearance of nuclear membrane, reappearance of nucleolus, and there is also the reappearance or resynthesis of endoplasmic reticulum and Golgi complex. So now each daughter cell again produces two daughter cells. So totally we get four haploid daughter cells at the end of meiosis 2. So this is the overall summary of this meiosis 1 and meiosis 2. So which brings about the formation of gametes in the gametogenic cells, or you can also call it as the germinal cells. Now you should understand the final part of it. What is the significance of meiosis? So meiosis conserves the chromosome number of each species by maintaining a constant ploidy level at fertilization. So the meiosis is going to conserve the chromosome number of each species. Okay, and it maintains constant. So the meiosis causes genetic variation. There is recombination of genes, isn't it? So that genetic variation leads to uh, crossing over in the, so due to crossing over and chiasmata formation, so there is genetic variation, which leads to variation in the population of organism. So which is an important aspect of evolution. Okay, these are all recombined chromosomes. You can see the red and white one. So they have all recombined in different ways. Okay, so this causes the recombination or genetic variation is responsible for evolution and an important raw material for evolution. Okay, so that completes our topic on the discussion of meiosis, uh, especially meiosis 1 and meiosis 2, along with significance of meiosis. So you have a